Reproduction is next. We'll also combine reproduction with human development. So talk about human development and pregnancy over here. We'll go through male anatomy. We'll do a little bit of introduction to meiosis, and then also go through the female reproductive system. Always going to look at the bird's eye view. Honestly, there's always higher levels of thinking and mastery level things to consider, but reproduction is a little bit lighter than other topics we've covered in AP2. So there definitely are some mastery level things that we can do, but even just looking at the levels of content, you can see that we're not quite high up on Bloom's taxonomy on this lecture. It's going to be more lower level knowledge and understanding. Quick intro slide that to me connotates the beauty of the human body. And then we'll go through the male reproductive system. So we'll start with the male reproductive system. Note the major structures highlighted in orange, and we'll go through those now. A common theme throughout this class is that cancer arises when cells stop following the normal rules of division. So the normal rules are don't divide when you're touching other cells because there isn't enough room. Don't divide when the DNA is damaged. And don't replicate when a cell is too young. Tissue that's really dividing fast, mainly epithelia, are more prone to cancer. And while the testes are not epithelia, they are producing sperm very quickly. So to have control of the speed of production of it, testes can control their temperature by changing their proximity to the body. For one, they're outside of the body to keep them cooler, but they can also be drawn closer to the body to keep warmer. So another problem with testicular cancer is that the immune system is isolated from the testes. This makes sense because sperm are a genetic mix of the host. We'll talk about this shortly. So since the sperm are different than the host, sperm are isolated from the immune system, otherwise the immune system would see them as different. This also means that cancer cells cannot be caught by the immune system as readily, so this is why testicular cancer is a common type of cancer. Sperm begins in the seminiferous tubules, these tubes down in here. Seminiferous tubules group into lobules. So these would be lobules. Those lobules come together into the rete testes. The rete testes carry sperm to the epididymis. Here's the head of the epididymis, and you can follow the pathway up to the body of the epididymis, and then the epididymis will carry up through the spermatic cord, up where sperm will basically be stored. At this point, sperm are not motile. They need to be activated by excretions of the prostate gland first. On the right side is a nice picture of the inside of the seminiferous tubules. And you can actually see small little sperm in here. So sperm begins on the outer layer of the tubule, and as they mature, so outer layer would be here, as they mature, they move inward. They're undergoing meiosis at the same time, which is why this figure is here. We're going to cover that shortly. Essentially, meiosis cuts the chromosome number in half so that when sperm and egg meet, they get back to the normal 46 chromosomes. Meiosis also mixes up the DNA, so offspring will be genetically different than the parents. Again, meiosis is depicted on the left, but we'll cover that soon. I don't know that there's a lot of really important anatomy in the sperm. The acrosome at the top contains enzymes that will help the sperm digest its way into the egg. Also, note the stacks of mitochondria that are in the tail to make sure there's ATP to provide motility. Cool picture. For the record, it's a misconception that the first sperm to get to the egg is the winner. Rather, the first few sperm at the egg will start to digest their way into the egg, but will not make it in before dying. It's the later sperm that penetrate the egg that has already been digested a bit. So it's not a case of the early bird gets the worm, it's a case of the second mouse gets the cheese. Or, often we tell ourselves that we were the one that won the race, but we were not the one that won the race. We were not the fastest sperm. Glands. We'll go through the various glands that support sperm next. At this point, sperm are stored in the ampulla, which is here in kind of a pinkish color, and that lies posterior to the bladder. Next to the ampulla is our first gland, the seminal vesicle. Again, the seminal vesicle lies on the posterior wall of the bladder and secretes about 60% of the volume of semen. Semen is a viscous, so thick, alkaline, basic fluid containing fructose, ascorbic acid, coagulating enzyme, and prostaglandins. Fructose and ascorbic acid provide nutrition for the sperm. Coagulating enzymes help sperm kind of stick together in the acidic environment of the vagina. And prostaglandins signal the uterus to undergo peristalsis. It actually moves or contracts in unison or in waves to help sperm make the journey to the egg. Sperm and seminal fluid mix in the ejaculatory duct and enter the prostatic urethra during ejaculation. Prostate, here in yellow, is a donut-shaped gland that encircles part of the urethra. So the urethra is the tube going through here. Ejaculatory duct would be here, coming from the ampulla and the seminal vesicle. The prostate produces a milky, slightly acidic fluid which contains citrate, a form of fuel, and then also enzymes. The prostate actually activates sperm to cause them to become motile, to become swimmers. A problem with the prostate is that it continues to grow throughout life. So normal, and here's enlarged. That's why as men age, they'll have to use the restroom more frequently as the prostate pushes up into the bladder and makes the bladder smaller. And also the urine stream will honestly be lessened too, because the prostate will push in and constrict on the urethra. A difficulty in diagnosing prostate cancer is deciding whether an enlargement of the prostate is due to this normal growth or is it cancerous growth. So the diagnosis of prostate cancer can be a bit of a gray area. It's one of those interesting things where medicine changes over time. The diagnosis of prostate cancer 10 years ago was a lot higher than it is these days because of this new recognition that the prostate is always growing, and so there's a difficult line to draw between whether something's cancerous or it's normal growth. Next is the bulbourethral gland, or sometimes it's called the Cowper's gland. Shown right here, it's pea-sized, sits inferior to the prostate. Bulbourethral gland produces a thick, clear mucus prior to ejaculation that neutralizes traces of acidic urine in the urethra. Sperm are pretty delicate, and so they need to be protected from everything, including the small amount of urine that might still be in the urethra. Next up is the penis, and we're not going to go into this into a whole lot of detail. I am more of a physiology person, and there isn't a whole lot of physiology to the penis other than that blood fills and is trapped in the penis to cause an erection. Drugs like Viagra target the vessels in the penis and cause vasodilation. As a review then for the test, I'd like you to know the basic A&P of sperm production. So specifically know the function and location of these structures: seminiferous tubules, retestes, epididymis, ductus deferens, ampulla of the ductus deferens, that it stores sperm. The seminal vesicle makes semen. The prostate gland activates sperm. The bulbourethral gland creates mucus to clear the urethra, ejaculatory duct, and urethra. Just the basic anatomy. Hormonal control. Know that essentially sperm production is controlled by the hypothalamus. So the hypothalamus would be up here, and then this would be the pituitary. It's controlled by the hypothalamus through hormones called follicle-stimulating hormone, abbreviated FSH, and luteinizing hormone, abbreviated LH. The interesting thing about those terms is they actually come from female anatomy, and you'll see that soon. I also put this information on this large infographic on the female reproductive cycle. I like to put it on here to contrast the difference between male and female hormonal control. So males are pretty simple, females are pretty complex. The gist of it is follicle-stimulating hormone and luteinizing hormone are released by the anterior pituitary. Follicle-stimulating hormone stimulates something called Sertoli cells to produce androgen-binding protein, a protein that holds and protects testosterone. Luteinizing hormone stimulates Leydig cells to make testosterone. Once androgen-binding protein has enough testosterone from the Leydig cells to stimulate seminiferous tubules to produce sperm, then they'll release that testosterone. So ABP concentrates testosterone made by the Leydig cells. Together then with another hormone called inhibin and an increased concentration of testosterone, that can signal back to the pituitary to decrease the production of FSH and LH. So what we have here is a nice homeostatic balance that causes continuous sperm production without over or under producing it. There's a nice video on hormonal control. Well, I don't know if it's nice. I'd like to believe it's nice. And we'll talk about that in a little bit, but you could get to it on the Prezi in that link. We're going to pause a little bit on reproductive anatomy to go into this process of meiosis. So before going on to female production, let's take a moment to talk about meiosis. We'll also come back to meiosis when we're talking about genetics, but meiosis occurs as sperm and egg are produced. That's why we're talking about it now. For sperm, it actually happens as the sperm move from out to in the seminiferous tubules. Meiosis accomplishes two tasks. It reduces the chromosome number from half, which is 2n, to n. So a normal cell has a pair of 23 chromosomes. So 23 is the n number. We have a pair of each of those, so we call that 2n. So we have 46 chromosomes. A sperm and an egg need 23 chromosomes so that when the sperm and the egg meet, you end up with 46 chromosomes again rather than 92. Meiosis also introduces genetic variability. We'll talk about this when we get to the genetics, but essentially genes can be swapped between a pair of chromosomes to mix up the genes even more. It's kind of shown here in this figure that we've got a little bit of the orange chromosome on the purple and a little bit of the purple on the orange. The reason we want this swap, for example, is hair color and eye color tend to have a pattern where brunettes have brown eyes and blondes have blue eyes, and that's because hair color and eye color are on the same chromosome. We don't always have this condition, so while that's the dominant pattern, there are exceptions, and those exceptions are due to swapping genes between the chromosomes. So once in a while you get a swapping of a chromosome, so you align blue eyes with brown hair. Maybe the purple chromosome here is brown hair, and the little tip of orange is blue eyes. Or maybe we should go back to here, that this chromosome here has brown hair and brown eyes. The orange has blonde hair and blue eyes. Brown hair is dominant, but you've got the blue eyes as well. So you've mixed up brown eyes or mixed up eye color and hair color because of this crossing over that occurs in meiosis. To allow the swapping of meiosis, you first have to duplicate the chromosomes up to 92. Then there has to be 2 splits to get down to 23. The steps of splitting those cells are the same as what you saw in mitosis. Do you remember the steps in mitosis? I like to remember them as IPMAT or I passed my anatomy test. That's an easy way to remember the first letters of each phase, so interphase, prophase, metaphase, anaphase, telophase. The first split from 92 back to 96 is called meiosis 1. Splitting again down to 23 is called meiosis 2. There's a little more detail in this slide. So when homologous pairs of chromosomes duplicate to get to 92, this is called synapsis. And the 4 chromosomes together form something called a tetrad. Tetrads allow crossing over, so the exchange of genetic material among tetrads occurs during synapsis. This is a nice figure to contrast mitosis from meiosis. In mitosis, adult cells are replicating and 2 identical daughter cells result. So if a cell dies in the liver and you need another liver cell, you go through mitosis to make an identical daughter cell. In meiosis, you end up with cells with half the normal number of chromosomes, and the DNA has been mixed up a bit. Again, the steps are the same in mitosis, and they look quite the same. So the DNA winds up to allow easier division of prophase. The chromosomes line along the equator in metaphase, so this was called the Metaphase Plate. When the chromosomes start to separate, we're in anaphase. I remember that because they kind of look like A's, and anaphase begins with A. When the cells divide, we're in telophase, and cytokinesis is the actual cleavage for, or the cleavage between the cells. So cyto means cell, kinesis means knife, so it's the knifing of the cell. Do note the crossing over happens in metaphase. As I mentioned earlier, crossing over mixes up genes so that genes that are normally on the same chromosome, like eye and hair color, can split up. So while brunettes usually have brown eyes, crossing over does allow some with brown hair to have blue eyes. So my dad has brown hair and blue eyes. I do guess I'm assuming here that Lauren Graham doesn't dye her hair or isn't wearing contacts to make her eyes look blue. We complete meiosis with meiosis 2, where the cells from meiosis 1 again go through prophase, then metaphase, line up on a metaphase plate, anaphase, where they kind of look like A's, and then telophase. In males, 4 sperm are produced. In females, 1 egg is produced, and the scrap chromosomes, that's kind of a horrible way of saying it, are put into a structure called a polar body. Let's wrap up meiosis with a review. So meiosis accomplishes 2 tasks. It reduces the chromosome number by half, 2n to n. So a normal cell has a pair of 23 chromosomes for a total of 46. A sperm and egg need 23 chromosomes so that a fertilized egg gets back up to 46 chromosomes rather than having 92. And meiosis also introduces genetic variability through crossing over. So that's meiosis. Again, we'll come back to that in genetics. I really did take a deep breath there. Deep breath. Let's go into female reproduction now. As a father of 2 girls and a husband, I already know don't even fake it. It's very complex. There's a lot of cool complexity here when it comes to the female reproductive cycle. So let's start with the anatomy. The ovaries are the primary female reproductive organs producing the egg itself, as well as the driving hormones, estrogen and progesterone. Accessory ducts, we'll go through some figures here in a second. Accessory ducts include the uterine tubes, the uterus, and the vagina. We can also divide the genitalia into internal, which would be the eggs and internal ducts, and the external, which would be the external sex organs. So let's start with the ovaries. Females have 2 ovaries that are intermediately positioned, so off to the sides, not in the middle, not really lateral. There are 3 primary types of cells in the ovary. There's the egg, which is in the middle, or it's called the oocyte, follicle cells that surround the egg, and then the granulosa cells, which follow the follicle cells or surround the follicle cells. When there's multiple layers, that's when we get these granulosa cells. About 7 million oocytes are in the ovary when a fetus is 3 months old in the womb. They decline throughout age. Even when you're born, a female will still have over 1 million oocytes. Primordial follicles are the beginning stage of the oocyte. About a year before an egg will be ovulated, it begins to develop into a primary follicle. Each month in a mature female, about 6 to 8 oocytes will develop into secondary follicles, and that will follow a pattern of basically every 2 months because the ovaries alternate in ovulation. So an ovary will produce 6 to 8 cells, oocytes, every 2 months. From secondary follicle, eggs will continue to develop to a tertiary follicle. From here, 1 egg will be selected, and that's for the most part. Occasionally, a couple of cells will be released, and that's how you get fraternal twins. When 6 to 8 tertiary follicles begin to develop, they're being supported by hormones from the anterior pituitary, hormones called follicle-stimulating hormone, we heard those, and luteinizing hormone. We heard those in the male anatomy. These cells are in a race to make their own estrogen so that they can continue their own development. So the cells need estrogen, and they're in a race to start developing their own estrogen. Once one cell begins to make estrogen, this estrogen signals back to the anterior pituitary to stop releasing the hormones FSH and LH. When the release of FSH and LH ceases, the cells that are not making estrogen are no longer supported, and so they wither. This is the mechanism that selects a primary follicle that moves on to a secondary follicle. So whichever egg can make estrogen first will cut off the growth of the other eggs. So the first egg to get to development first basically cuts off the control, or cuts off the ability of the other eggs to keep on maturing. When the tertiary follicle is mature, it will be ejected in a process called ovulation. So we're ejecting this oocyte in ovulation. The granulosa cells that remain behind form something called the corpus luteum. The corpus luteum will be very important in the first weeks of pregnancy, as the corpus luteum makes the estrogen and progesterone necessary to maintain the pregnancy. So these hormones will prevent a continued cycle of the uterus. And so they're very important in order to maintain the pregnancy. Figures rarely show this, but the corpus luteum will greatly enlarge, reaching a size almost the size of the ovary itself. It'll turn yellow as it accumulates cholesterol. And why cholesterol? Because cholesterol is the precursor for estrogen and progesterone. For more details on this process, refer to the infographic and the YouTube on the female reproductive cycle. We'll do fallopian tubes, uterus, and vagina next. From the ovary, eggs will pass into the uterine tubes, which are also referred to as the fallopian tubes. The area of the uterine tubes that surrounds the ovary is called the ampulla. And the ampulla has projections, or finger-like projections, called fimbriae that wrap around the ovary. There's a space between the ovary and the fallopian tubes, and that there's not a whole capsule around the ovary. So this means that eggs can miss the fallopian tubes and can still be fertilized. Also, cells that normally line the uterus can find themselves outside of the uterus. Unfortunately, these cells might still go through a menstrual cycle and release blood into the abdominal cavity, and that blood causes pain when it's in the wrong place. So this condition is called endometriosis and is really painful. Somewhat relatedly, if there's bleeding during ovulation, this can also cause a pain called Mittelschmerz, which some women can feel when they're ovulating. In this image, we can review again the ovary, the ampulla, which is this area here. The fingers are the fimbriae. And notice that there's an opening between the ovary and the fimbriae. So the egg can escape this way and still be fertilized, which would lead to an ectopic pregnancy. Or also sometimes cells from the uterus can find themselves outside the uterus, still menstruate, release blood, and that blood causes pain, and that condition is called endometriosis. One more term is the end of the ampulla. It's called the infundibulum. The top of the uterus is called the fundus, and the main part of the uterus is called the body in yellow here. The isthmus is the narrow tunnel down here that connects the uterus with the vagina via the cervix. The cervix is right here, the opening into the uterus. The cervix is the narrow opening between the uterus and the vagina. Repetitive there, I guess. The cervical canal is the length of the area around the cervix, so through here. The opening on the side of the vagina is called the external os, and the opening facing the uterus is called the internal os. I always find this interesting that the cervix is blocked with mucus on most days of the cycle to prevent pathogens from entering. The opening is only cleared around the time of ovulation, so sperm can enter and get up to the egg. Here's the larger infographic and a link to the video. Here I'd also like to point out that this is the Corpus Luteum, and it's almost the size of the whole ovary, and it's very yellow because of the colostrum. Hormonal control. I might recommend watching this video to have a big picture understanding of the full female reproductive cycle, or as best as I can do. We'll still go through it in the next few slides though. So first, a female reproductive cycle is generally about 28 days. The length of the cycle will vary for some women, but it's typically 28 days. My wife, maybe I shouldn't say this, but it's really short. It's like 20 days. Days 1 through 14 are when the follicle is developing. That's called the follicular phase. Day 14 is ovulation. Recall that after ovulation, the cells that remain behind are called the corpus luteum. Since the corpus luteum becomes the director of the cycle, this phase is called the luteal phase. Here it is just visual form. Follicular phase, ovulation, driven by the corpus luteum, so this is called the luteal phase. There are a couple of errors in this figure, or at least differences from my understanding. So one of the things is it's the tertiary follicles that actually compete, not the secondary. And also, it's my understanding that we go from primordial to primary to secondary about a year before ovulation. It's not just 2 months before ovulation. I think what they're trying to say is the ovaries alternate every 2 months, and that's how you get 1 egg per month from 2 ovaries. But it does show down here some nice things where you've got the follicular phase, ovulation, and the luteal phase. We've covered ovulation already, so ovulation is usually at day 14 of the cycle, and we've mentioned Mittelschmerz, and we've mentioned fraternal twins because of overovulation. After ovulation, the ovary enters the luteal phase. The corpus luteum will continue to generate estrogen and progesterone. If an oocyte is fertilized and implants in the wall of the uterus, it'll send a signal back to the ovary called human chorionic gonadotropin, or HCG, to signal the corpus luteum to stick around. HCG is what is tested for to determine pregnancy. If there's no implantation and no HCG, then the corpus luteum will degenerate in 10 days and leave a scar called the corpus albicans because it turns white. Albicans is white. Here's a nice figure from Wikipedia, but I like the figure that has everything in it with some description. So that's why I created an infographic, which I pitched multiple times already. Did I mention I have an infographic on this? Just kidding. Kind of, but not really. I like it. We covered the ovarian cycle before, but let's look back at the uterine cycle. Because the beginning of the menstrual flow is the most overt or outward sign in the ovarian or uterine cycle, the beginning of the menstrual bleeding is designated as day 0. So shedding the lining is dead 0. Days 1 through 5 involve shedding the lining, leading to menstruation. At days 6 through 14, the uterine wall is built back up in the proliferative phase. And recall again that at day 14, that's when the egg is being released. It's going to take 6 to 7 days to actually reach the uterus. So that's why the uterine wall doesn't have to be built quite all the way up until day 21. So the egg is ovulated on day 14, but the uterine wall continues to develop in the secretory phase, so secretory phase, as the egg generally takes 6 days to travel down the uterine tubes. So the secretory phase is from days 15 to 28. Let's take a little tangent here and go into estrogen. Estrogen levels rise during puberty. Estrogen promotes oogenesis and follicular growth. It exerts anabolic, so building effects on the female reproductive system. So uterine tubes, uterus, and vagina grow larger and become functional. Uterine tubes and uterus exhibit enhanced motility, and the vaginal mucosa thickens and external genitalia mature. It also induces secondary sexual characteristics, things like growth of breasts, increased deposition of subcutaneous fat, especially in the hips and breasts, widening and lightening of the pelvis, and growth of axillary, so armpit and pubic hair. That's hormonal control. When talking about estrogen, we should also talk about the connection between estrogen and breast cancer. Cancer usually arises from the epithelial cells of the ducts. Risk factors include early onset of menses, so the beginning of the cycle early on, or late menopause, no pregnancies or first pregnancy late in life. The reason for this one is that estrogen is thought to promote cancer, but progesterone is thought to offset the effects of estrogen. And progesterone levels rise considerably during pregnancy and offset what are also increasing levels of estrogen. So the fact that there's more progesterone during pregnancy to protect you from the estrogen is why pregnancies are thought to influence breast cancer. Previous history of breast cancer, hereditary factors like BRCA1 and BRCA2. 70% of women with breast cancer have no known risk factors. That's kind of scary. And 95% of breast cancer is non-heritable. It's kind of scary. These are more scary facts. 1 in 8 women will develop breast cancer, and 1 in 35 women will die of breast cancer. That last statistic is one of those where you're lucky enough to live long enough to get cancer, or it's kind of like Alzheimer's. Most people that are over 90 will develop signs of Alzheimer's. So if you're lucky enough to live long enough, that's when you get Alzheimer's. It's a weird way to put it, but it's kind of true. Early detection of breast cancer is accomplished by self-exam and mammography. It's interesting how mammography has changed in the last few years because there's a recognition that the radiation from mammography can also create a risk. Treatment depends upon the characteristics of the lesion, so radiation, chemotherapy, surgery followed by irradiation and chemotherapy. Today, lumpectomy is probably the surgery used rather than radical mastectomy. That could be one of the mastery level things that you think about, the way that Angelina Jolie just had her breast removed to prevent the risk of breast cancer. So now we're going to move into pregnancy and human development. Very cute baby. Fertilization. Some terms here first. So pregnancy, events that occur from fertilization until the infant is born. Conceptus is the developing offspring. Gestation period is from the last menstrual period until birth. Pre-embryo is the conceptus from fertilization until it's 2 weeks old. Embryo is conceptus during the 3rd week through the 8th week. At 8 weeks, all the organ systems are there, and so the embryo becomes a fetus. So fetus is conceptus from the 9th week through birth. There's some interesting math here. The oocyte is viable for 12 to 24 hours. Sperm can survive in the uterus from 24 to 72 hours. This means that fertilization can occur if coitus occurs or sex occurs 3 days before ovulation or 24 hours after ovulation. The term fertilization is used for when sperm meets egg to form a zygote. This is an interesting graph from an old article in the New England Journal of Medicine. If coitus occurs 2 days before ovulation, the likelihood of pregnancy is around 35%. It's over 30%. I think this is interesting because I think most of us think getting pregnant is difficult. We've all heard of somebody that's had difficulty getting pregnant or conceiving, and so we think that getting pregnant is difficult. So it's a matter of hearing about the stories of people who have had difficulty getting pregnant, but a lot of people have no problem getting pregnant. Let me tell you my story. Just kidding, sort of, but both of my kids were conceived when we just weren't trying quite as hard to not get pregnant. And my first one, we actually got pregnant without even knowing it, and we didn't know we were pregnant until we were 4 months along. So first kid, it's like 5 months. You're gonna have a kid 5 months from now. Didn't get in extra games of golf or anything like that. That you should get in if you know you're gonna have your first kid. I know why. If you have coitus twice or 3 times in the 6 days before ovulation, a pregnancy rate of 56 to 57% occurs. Predicted rates is a little bit higher, but you're more than half as likely to have a pregnancy if you have sex twice during the 6 days before ovulation. Humans are good at reproducing. An ovulated oocyte is encapsulated in cells called the corona radiata, which extends fingers. We're going to see this in the next picture. The layer of cells under this layer is called the Zona Pellucida. Sperm binds to the Zona Pellucida and undergoes the acrosomal reaction where enzymes are released near the oocyte. I mentioned this before, that it's not the action of the single sperm that penetrates the egg. It's It's got to be hundreds of sperms and their acrosomes to release the enzymes necessary to digest the Zona Pellucida to allow a sperm to finally penetrate the egg. So corona radiata, it's kind of like the sun has a corona radiata. So radiating rays, I think is what that means. That's the corona radiata. Zona Pellucida is where the sperm actually try to bind to, and the acrosome will start to eat through the corona radiata so that a sperm can penetrate into the egg. Kind of a cool picture, totally fake from Wikipedia. But again, remember, the first sperm isn't going to win. It's the later swimmer that's able to penetrate the Zona Pellucida that was weakened by the sperm that came before them. Once a sperm makes contact with the oocyte's membrane, the oocyte pushes the Zona Pellucida away, so no more sperm can enter the cell. So basically know that there's specific docking reactions here between the egg and the sperm. And once there's a docking, the docks are withdrawn, so other sperm can't penetrate the egg. This is kind of a fun game, and it's sort of physiologically accurate. So look for things like, if you play it, look for things like that the cervix is policed by white blood cells. I mean, a sperm is a foreign object, and it can be killed by white blood cells. And the uterus will contract on the side of the ovary that's ovulating to help sperm reach the egg. It's kind of a good study break, too. Let's now follow the events after fertilization. So once the DNA of the sperm and the egg mix, the egg is termed a zygote. It then begins to divide as it moves down the uterine tube. This figure begins the division, but the next figure is a little bit more specific. So 2, 4 cell, 8 cells. This one's more specific because it shows an unfertilized oocyte, fertilized oocyte, 2 cell, 4 cell, 8 cell morula. I don't think you need to memorize all these. A fertilized cell that begins to divide is called a blastomere. Then it goes to a morula, so this would actually be the blastomere. From a morula, it then becomes a blastocyst. A blastocyst is the stage where implantation occurs. So when embryos are created for an in vitro fertilization, they're usually halted at the 16-cell stage, so before the egg is even ready for implantation. That's fertilization. Once the blastocyst sticks to the wall of the uterus, it becomes surrounded by uterine endometrial cells. So it becomes surrounded by the uterine cells. Implantation is complete at 14 days after fertilization. So in the images, green, blue, and purple cells form the blastocyst. Cells in the uterus change their morphology to support the implanted blastocyst, and these cells will become the placenta. These cells are also there to signal back to the corpus luteum that the pregnancy has been complete. So when the trophoblasts here develop, they start releasing human chorionic gonadotropin, or HCG, and that's what goes back to the corpus luteum to tell it to keep secreting progesterone and estrogen to maintain the pregnancy. HCG is also what's picked up in pregnancy tests. This is also why you can't know that you're pregnant for at least 6 to 10 days after fertilization, because you have to wait for the egg to actually implant. You have to wait for the trophoblasts to start releasing HCG. The corpus luteum will then release progesterone and estrogen until about 12 weeks. At that point, the placenta will take over producing estrogen and progesterone necessary to maintain the pregnancy. Here, the concept is just represented in a more visual form. So HCG is released by the trophoblast, which tells the corpus luteum to continue making estrogen and progesterone. Estrogen and progesterone will maintain the endometrium and the pregnancy by preventing further ovulation. So recall how estrogen inhibits the anterior pituitary from releasing the hormones that cause follicles to mature. Those hormones are FSH and LH. So by continually producing estrogen, you're going to inhibit FSH and LH, and the cells won't develop, so you won't have primary follicles growing up at this point. The placenta will eventually take over the production of estrogen and progesterone at about 12 weeks. Sorry, I like representing things graphically. So this graph shows how before 12 weeks, estrogen and progesterone, shown in orange and blue here, are produced by the corpus luteum. After 12 weeks, their production ramps up quite a bit, but this estrogen and progesterone is created by the placenta. Eventually, the estrogen levels get so high that that's actually the signal for the uterus to contract and begin labor and delivery. You certainly don't need to memorize all these details, but you might have heard the term germ layers. Beginning at the blastocyst, cells become determined. That is, they're limited in what they can become further. So, for example, cells that would become internal organs down here come from what's called the endoderm. The mesoderm becomes the circulatory system and lungs, and the ectoderm becomes the skin and the nervous system. So once you've reached past the determined layer, a cell is kind of in one of these buckets and it can't be something else. I also find it really curious that cells that are in the ectoderm are going to become either the most complex cell in the body, so a neuron, or one of the most simple cells in the body, the simple squamous cells that make skin. The germ layers are then going to fold, so you can see why the endoderm will become the GI tract. So the endoderm will fold and become the internal structures and the gut. The process of developing the organs of the body is called organogenesis. So prior to 8 weeks, the conceptus is called an embryo. An embryo does not yet have even the beginnings of all the organ systems. At 8 weeks, all the organ systems are present, though there's considerable more development that is needed. Once all the organs are present, the embryo becomes a fetus. Kind of cool picture of an embryo. Female changes. There's some anatomical changes that occur in pregnancy. So Chadwick's sign describes how the vagina develops. A purplish hue, breasts will enlarge, the areola will darken, the uterus expands, occupying most of the abdominal cavity. Lordosis or swayback is common due to the change of the body's center of gravity and kind of a, oh Lord, I'm pregnant. Relaxin causes pelvic ligaments and the pubic symphysis to relax. Typical weight gain is going to be about 29 pounds, with too much more or too less than that often affecting the health of the newborn. This figure shows how the uterus pushes up into the abdominal cavity. Maybe think about the times you've been viewing the cadaver and thought how things are just really packed in there so efficiently. Now imagine adding an 8-pound baby and 20-odd pounds of other tissue and fluid. It's a lot to squeeze in. Pregnant woman also undergoes several physiological changes. The placenta secretes human placenta lactogen, which basically helps with the production of milk. HPL promotes growth of the fetus and exerts a maternal glucose-sparing effect too. So the woman, the female, the mother doesn't use the glucose, it's spared for the baby. Human chorionic thyrotropin, which increases maternal metabolism. Parathyroid hormone levels are high, ensuring a positive calcium feedback. Are you noticing that I didn't want to pronounce that? Somatomamotropin. The organ systems change as a whole, so the GI tract responds to the increased estrogen and progesterone, and that's thought to cause morning sickness. Urinary system increases urine production because there's an increase in handling of the additional fetal waste. Respiratory system might become edematous, and the nasal congestion may occur due to the extra fluid being carried. So the fluid in the lungs and the baby pushing up into the diaphragm can also lead to difficulty breathing. Continuing, the cardiovascular system sees 25 to 40% increase in blood volume. Venous pressure from lower limbs is impaired and may result in varicose veins. Labor and delivery. There's 3 phases: initiation, dilation, and expulsion. As we've said earlier, estrogen continues to rise. So estrogen was in orange throughout the pregnancy and reaches a peak during the last weeks of pregnancy, causing myometrial weakness, so weakness of the uterine muscle, and irritability. This irritability sets off weak practice contractions called Braxton-Hicks contractions, and those often occur. So as birth nears, oxytocin and prostaglandins cause uterine contractions, as well as emotional and physical stress, as well as activities of the hypothalamus. This sets up a positive feedback where more and more oxytocin is released to further stimulate uterine contractions. So we've talked about this before because it's the classic example of positive feedback. So the cycle is that the head of the baby pushes against the cervix. This stimulates the posterior pituitary to release more oxytocin. So the head of the baby pushes against the cervix. Nerve impulses are transported to the brain, tells the posterior pituitary to release more oxytocin. When you release more oxytocin, it goes through the blood down to the uterus and stimulates more contractions. When you stimulate more contractions, the head of the baby pushes harder against the cervix and causes the release of additional oxytocin. Nice, gentle, positive feedback that builds and builds and builds the strength of contractions until the baby is delivered. After labor is initiated, the next stage is dilation. This stage continues from the onset of labor until the cervix is fully dilated, the classic 10 centimeters. So initial contractions are 15 to 30 minutes apart and 10 to 30 seconds in duration. The cervix turns outward or effaces and dilates. The amnion ruptures, releasing amniotic fluid. This is called breaking the water. And then engagement occurs as the infant's head enters the true pelvis. So we're at 10 centimeters and the infant's head is inserted into the pelvis. Next is the expulsion stage, which takes place from full dilation to delivery of the infant. Strong contractions occur every 2 to 3 minutes and last about a minute. The urge to push increases if there's not local anesthesia, and crowning occurs when the largest dimension of the head is distending the vulva. Here's a nice graphic of expulsion showing the stages of presentation of the head, rotation and delivery, delivery of the posterior shoulder, delivery of the lower body and umbilical cord. Over the next 30 minutes, the placenta is delivered as well. Afterbirth is the term for the placenta, and it's attached to fetal membranes. All placental fragments must be removed to ensure no part of the placenta is still attached to the mother's circulatory system. If fragments remain, severe postpartum bleeding is possible. A nice picture of how the placenta collapses and is expulsed. Postpartum. Once the baby's born, at both 1 minute and 5 minutes, the baby's health is scored using the APGAR score. APGAR is based on skin color, pulse, reflexes, muscle tone, and breathing. A score of less than 8 is deemed a concern. It's kind of a classic. It was actually named after a Dr. APGAR. As we've learned in AP2, the will to breathe is generated by carbon dioxide. So as carbon dioxide builds up, the urge to take that first breath is initiated. This takes a lot of effort as the lungs are sort of stuck together and air needs to open up the air sacs of the alveoli. This also is what's happening when you get the air knocked out of you. First 68 hours is unstable, vacillating back and forth between wakefulness and sleep. This is the last time my Celia looked like an angel. I'm just kidding. She's got energy. In this transitional period, activity cycles in roughly 3 to 4 hour periods of activity and rest. And the baby will start to sleep through the night, but man, my beautiful Celia didn't sleep through the night until she was at least 2 years old. Still is up most nights. Generating the machinery to produce milk is complex. Many hormones play a role. Estrogen, progesterone, lactogen, and prolactin ensure milk production. And then the hormone oxytocin, which we'll cover in a second, actually readies the breast to deliver milk in a process called letdown. The initial milk for 2 to 3 days is especially rich in vitamins, minerals, proteins, and antibodies, and is called colostrum. You can buy it in a store if you like, and even in open-air markets in some countries. You can buy it in pill form in the States. It's bovine colostrum, though. Suckling of the baby, or even the sound of a baby crying, not even that mother's particular baby, can stimulate milk letdown to prepare the breast to release milk. If we read through the slide, we can see that there are many benefits of breast milk. So fats and irons are better absorbed. Amino acids are metabolized more efficiently than those of cow's milk. Beneficial chemicals are present, so antibodies, other immunoglobulins, complement, which is for the immune system, lysozyme, which will kill bacteria, interferon, which will kill viruses, interleukins, prostaglandins, which will stimulate white blood cells and prevent inflammation. And then there's natural laxatives to cleanse the bowels of meconium, that first bowel movement. My wife, I want to always caution this. I always feel like I should say this. Though on a personal note, it isn't always possible to breastfeed. My wife couldn't produce enough milk. It was a very difficult couple of days because Ella, our first child, continued to lose weight and wasn't getting enough food, and so was basically crying all the time. So we eventually had to move over to formula. So as you read these advantages, realize they're the best case scenario. But if it doesn't happen for you, your kid's going to be smart, wonderful, and beautiful, even if they don't get breast milk. For some mastery level thinking, look into testicular cancer or prostate cancer. You might look into the evolution of prostate cancer and its diagnosis in the last 10 to 15 years, or look at some of the treatments too. It's kind of a nice, it's a different cancer to irradiate, so there's often different techniques there. So there's proton radiation. Now, I'm not really up to date on my particle physics in physics, But my understanding is that an X-ray is a smaller electron-like structure, whereas a proton, think about the structure of, think we're going all the way back at the end of this semester, going all the way back to chemistry and how you have protons and neutrons in the nucleus of an atom, and then you have electrons. Electrons are really small, so when you shoot electrons at things, it doesn't do a whole lot of damage, but if you can shoot protons at things, those are much bigger bullets, and so proton radiation shoots at bigger bullets at a cancer cell. Something to look at. Just again, the evolution and how wonderful humans are at coming up with different treatment methods. Explore why genetic variability is a benefit in humans. You could explore pathologies associated with female reproductive anatomy like endometriosis, other factors that are used to monitor breast cancer. You could look into the different guidelines for mammography these days. Again, I just think it's interesting when medicine evolves. Investigate the term linea nigra, which is a brown melatonin-based line that goes from the belly button down to the vagina in some women during pregnancy. Interesting phenomena. Again, why would pregnancy cause this increase in melanin in this just one little line from the belly button to the vagina? Investigate health issues associated with labor and delivery, or explore costs associated with childbirth by method and country. For that matter, you could look into the cost savings of breastfeeding your child. That's one of the reasons to breastfeed your child, is it saves a whole lot of money. All right, that's the reproductive system. Thank you.